Thank you very much for the opportunity to present, and also thank you for the Innovation Council for supporting our project with the SID grant. So I'll talk today about how to recondition or improve the quality of aged livers using the combination device, Normothermic Perfusion device, and stem cell activating biologics. Specifically, I'll talk about idea behind it and what it entails. So why aged donors are a challenge for us? So our population is aging very fast. If you look at the map, by 2040, over 20% of our population in the United States are going to be over 65 years old. And that compares to 4% of the same group of within the population in early 1900s. So the organs from older individuals, not only there is a concern for regenerative capacity, which there is for sure, we also increase comorbidities with aging, and that can also contribute to poor quality of the organs. So aging poses a significant issue in transplantation and donation because, as you can expect, over 91% of individuals who pass away are over the age of 65. However, if you look at the organs that we're using from the deceased donors, only 7% of our organs that come from deceased donors are from the donors over age of 65. And the reason for this, there is a reluctance of accepting older donors. There are multiple reasons. Again, older organs are susceptible to ischemic perfusion injury that leads to the poor graft function or actually primary non-function, decreased regenerative capacity and metabolic capacity, which is especially important in liver, increased immunogenicity, which means potential increased rejection. There are concerns for durability of those organs due to the pre-existing comorbidities such as fatty liver disease. And then also, if you're talking about living donors, if you take part of the liver for donation, you don't know how their own liver is going to regenerate due to their age. So what are the potential rehabilitation strategies? The first one that I explored was really in my early on with KO8 award using iPS stem cells to rehabilitate organs. However, then I met Dr. Helms and her collaborator, Dr. Liu, that have deep understanding of actually adult stem cell biology and specifically worked for many years on a Wnt pathway. Wnt is a molecule that in a certain condition can initiate self-renewal of the stem cells. However, under other conditions, it actually promotes differentiation and regeneration. In liver specifically, during the hypoxic injury, there is actually natural derivative of Wnt is being produced from the endothelial sinusoidal cells that eventually activates the adult hepatic stem cells and promotes hepatocyte proliferation and tissue repair. So our aim was to amplify this regenerative effect by administering highly stable synthetic biologic to the damaged livers ex vivo. So we presented some preliminary data in our ischemic perfusion model in the lineage tracing animals. We induced ischemic perfusion in one single hepatic lobe by clamping it for 15 minutes following the perfusion. We demonstrated that there is a target for the Wnt, specifically because the Wnt responsive stem cells increase in numbers at 7 days following this ischemic injury. However, it's not necessarily proliferation. They did not proliferate fast, so it's more the fact that the ischemic injury potentiates stem cells to Wnt responsive state rather than increased proliferation. So we also demonstrated that when preserve the animal liver, specifically the actual liver slices with Wnt biologic, we were in the normothermic temperatures, we were able to activate against Wnt responsive stem cells as well as decrease cell death due to the ischemic damage in the liver. So this biologic works best at the normothermic temperatures, and as well as it's extremely stable. This is specifically the biologic that was produced by Dr. Helms. So it has stability over 80% at normothermic temperatures for 4 days. So how do we administer this? What are the best options? Because we have to try to, first of all, give the time for that biologic to work in the organ in order to optimize it before putting it into the recipient, as well as minimize the potential systemic side effects of those biologics without administering it actually into the body. So these days, best option is utilization of machine perfusion devices that became very popular across the globe to preserve the livers. So there are normothermic oxygenated machine perfusion and there are hypothermic oxygenated machine perfusions that are available in the United States right now and across the globe. The two most common that we're using in U.S. is the Transmedics OCS. It's normothermic as well as the Organox. The other two kinds is hypothermic. They're either in the clinical trials or in a process of getting de novo 510 approval at the moment. So there are combination machines. However, this one is not approved for normothermic preservation right now in the United States. So therefore, our really target was trying to administer the Wind Biologic on normothermic machine perfusion, given its stability and activity at the normothermic temperatures. The other benefit of those machines, at least currently in clinic, majority of centers feel comfortable preserving the livers up to 24 hours at minimum. Actually before transplantation, so it gives you time to administer the biologic. But you also need to recognize that there are even further steps. You can take very extremely marginal organs. First, how does machine perfusion work in itself? So it's been demonstrated there is a common pathway. When the organs are being retrieved from the donor, over time there is an ATP depletion, and that actually contributes to organ dysfunction and damage. Both machines, hypothermic and normothermic, actually restore ATP levels at the time of the perfusion. And it's been known actually to correlate with improved outcomes following the transplantation. So again, as I mentioned, there are actually more extreme benefits of this machine. So this is an example of the Chinese group that took 80% macrosteatotic organ that we would never take, because just for your reference, 30% macrosteatosis is associated with primary non-function, high chance of primary non-function. They were able to perform ischemia-free transplantation where the liver is actually taken out of the donor directly onto the machine without subjecting the organ to any ischemia or damage. And following maintenance on the machine and optimizing the organ on the machine, they were able to transplant it into the recipient with a successful outcome. So the liver was maintained for 4.5 hours. In majority of our cases, we follow the lactates that improved significantly over the course of 1 hour, as well as the stable pH. So even more interesting, actually, Dr. Helm's colleagues from Zurich, under the leadership of Professor Clavey, and they developed a normothermic machine perfusion where organs can be preserved for extended period of time because they incorporated dialysis unit into the system. In this case, the clinical transplantation was performed after the liver was maintained in the machine for 3 days ex vivo. And the subsequent studies also demonstrated actual ability to maintain those livers for 5 days. So this really shows you how this technology allows the delivery and optimization of the organs. So our proposal, of course, delivering the L-Wind, the stable molecule, to the organs ex vivo in the machine after the organ comes out of the donor, and perfused over the course of several days. This with goal of reversing tissue damage as well as promoting tissue regeneration ex vivo. So we built the prototype of the perfusion machine in our lab. I have a great research assistant and medical student who worked on it by several months. They have the inline peristaltic pump, oxygenator with a flow meter control as well as manometer. The liver is actually sitting in the bath that maintains the normothermic temperatures of the perfusate and of the organ, and that really allows us to maintain those organs and also deliver delivered therapeutics. So we have the model that's now sustains the mouse model as well as the rat model, which is a little bit more complicated because actually it's more difficult to distribute the biologics equally throughout the entire organ of the rat. And we're still working on this. But I'm really thankful to our microsurgeons, Dr. Han and Dr. Tajima, for helping us with this. We demonstrated that our normothermic machine perfusion does not damage the organ. This is a control versus 1 hour of perfusion on our machine. And in parallel, we're also studying the biomarkers of the liver viability as well as therapeutic targets in the human livers. This particular study looked at human livers that were discarded for transplant. However, they were divided into 4 categories. Lean livers that had adequate hepatocyte function that actually considered to be viable. And the viability was studied based on lactate production as well as the pH control in the flows and additional other parameters, which is standard for monitoring perfused organs. Or also lean livers that were not actually considered to be adequate and non-transplantable, as well as the steatotic livers that both adequate function and inadequate function. What we demonstrated is that viable livers during normothermic machine perfusion, including both fat and non-fat livers, are demonstrating upregulation of markers related to the regeneration as well as metabolic fitness. So really that tells you that machine can help you to not only, number one, determine which organs will be functioning well in the recipient compared to the non-viable livers. But more interestingly, when we looked at the detailed subcategories, what we found that there are specific genes that are downregulated in non-viable livers, specifically in steatotic livers, and they're associated with the Wnt pathway. 2 of them, GRB1 and SORT1. So this actually may lead us further on studying this particular genes and this particular subcategory of livers in our lab on the animal normothermic machine perfusion and see whether or not Wnt and actually optimize the fatty non-viable organs there. So what are our future directions? We got extension funding from Biodesign after my faculty Biodesign fellowship, first of all to establish the prototype of the delivery system as well as getting additional consultation for regulatory and reimbursements strategies. We got additional funding from Lucine Packard to improve quality of kidneys for pediatric transplantation. Again, we hope to use our machine perfusion for this. And lastly, one of our wonderful first-year residents established a great relationship with the Genentech, actually utilizing this machine, applying selective caspase inhibitors for the organ regeneration. So I would like to thank first of all my Circle of Life Lab, my awesome members, all of my partners who support me throughout clinical and research and funding. Thank you so much.